30 and welcome to our genetic engineering video. We're going to get started talking about genetic engineering. So there is going to be this video linked in to our Edpuzzle here, and you're just going to watch it to get things started and then move on into the notes. So genetic engineering, what is it? So genetic engineering is essentially when we go in and we take DNA from one organism and we splice it together or cut and then put it together with another organism. Okay. When we do this, we refer to it as splicing. And when we are able to splice two things together, it changes the overall uh, genetic components of your DNA, and you express different genes. Now, they have used this kind of process for a long time. It's a big refinement on traditional breeding practices. Okay, so if you think about if you wanted to have a specific type of animal, you picked the best animal in your grouping and you bred it with the ones that you also thought had some of the best characteristics. Okay, that was called artificial selection, right? Picking two things and putting it together. But essentially, it's still the idea of genetic engineering. We're just making a refinement to the practice that it is. Okay. Now, what ended up happening was when people started looking at this, okay, it was because of technology that we figured out from cloning experiments. And now cloning comes from asexual reproduction, realizing that cells can make identical copies of each other. So the technology just kind of kept building and building upon each other. So in order to do genetic engineering, you have to have a couple of important things. The first thing you need to do is you need to get what is called a plasmid from a bacteria. So a plasmid is a circular shape inside the bacteria that can be inserted into different bacteria. But you can also insert new DNA onto it and it'll uptake that DNA. So for us, we're going to use the E. coli bacteria. And the reason we're using the E. coli bacteria is because it's cheap, there's lots of it, it's easy to manipulate, lives short, and it naturally lives in our gut. So it's okay to use, it's not super dangerous. We also are going to need a gene we would like to clone. So in the case of this example, we're going to need to make insulin. So we're going to try and find the human insulin gene, and we're going to then cut it and add it to our DNA. In order to cut DNA, we need something called restriction enzymes. Restriction enzymes are essentially the scissors. They're going to cut open the plasmid so that we can glue in the DNA. Restriction enzymes are a little bit they're still used, but they're still more of a thing of the past because we have some more recent technology that does a better job, and we'll talk about that here in a bit. But essentially, we're going to cut the DNA and then we're going to glue it in. And you guys know what ligase is because we've seen it before. But essentially, you're going to glue in the DNA that is needed for the process. So I've already kind of spoke about this. Why are we using E. coli? Again, there's lots of it. It's easy to make, it's cheap, it's easy to work with, and it naturally lives in our guts. So it's really important that we want to find something that's safe for us to use as well as easy to use. Now, if you look at the picture on the right-hand side, you'll see that it has its bacterial DNA, and then you'll see those circular plasmids. The picture below is just examples of isolated plasmids. So they're not exactly 100% circular, but they have a small circular shape, okay? And what we want to do with this small circular shape is use it to help do replication of the gene that we want. So plasmids can be replicated separately from the bacteria's DNA. Okay, so what ends up happening is it replicates on its own without actually affecting the genome or the bacterial DNA that is on the other side. So they're really, really handy because we're not going to actually harm the bacteria or change the bacteria. The plasmids will have this own replication on their own, and they will be able to replicate the gene we want without affecting the DNA. So this is a little bit about restriction enzymes. So restriction enzymes are to cut DNA at specific recognition sequences. So restriction enzymes have a specific way that they actually cut the DNA. So they look for a sequence that ranges from about 4 to 8 nucleotides. Remember, nucleotides are our A's, T's, and G's, and we call them palindromes. So what that means is when you have a palindrome, it is the same forward as it is backwards so that it looks, so it's easy to read. So if you see what's going in on the picture there, you'll notice it runs AGCT on the left side, but TCGA on the right side is AGCT, and on the left side is TCGA. If you read them backwards, they're the same thing. They're palindrome. So we have about 3,000 different ones, or 600 commercially available palindromes that we can use to help with, restriction enzymes that we can use to cut. Some come from bacteria, such as the from the EcoR1, which is the E. coli, and it is used for defense mechanisms, and it stops bacteria from gaining viruses. So it can actually selectively cut foreign DNA and cause that DNA to be ineffective. So EcoR1 is one type of restriction enzyme. So we're going to skip over the restriction enzyme BAMH1. We're not going to need to worry about that. Instead, we're just going to talk about the genetic engineering process. We're going to go into a little bit more detail. So we know the 4 pieces that we need, but now let's talk about the actual process. So step 1 is you have to isolate your plasmid from the E. coli bacterium, and you have to cut it open using the restriction enzymes. So isolate and then cut. From there, you need to isolate your gene from a chromosome that you're using from some sort of DNA, whether it's a human genome or something else, and you're going to use the same restriction enzyme and cut that desired gene out. You're going to place both of the plasmid and the gene in a test tube, and you're going to glue those pieces together. And then you're going to take your recombined plasmid. So recombined just means that it has now the plasmid and the DNA gene that you are wanting. And you're going to put it back into the E. coli so that it can start to replicate. So once it's in the bacteria on the Petri dishes, those plasmids can start to replicate on their own. So when the bacteria grows and mass produces, whatever protein or gene is inside of there will start to be made. And then proteins can be taken by being isolated, sterilized, and bottled, and then sold in pharmaceuticals industries. So that is how you essentially gain a isolated gene from a bacteria. So that's genetic engineering. If we were to look at the picture here, you can see here's my human cell, and I'm going to have my chromosome piece. Here's my bacterium, okay? I've sliced up my chromosome. I'm looking for that desired piece, that gene I want. Here it is. I've cut up my plasmid. I am now going to glue them together. Here's my bacterium I'm going to use, and I'm going to insert that plasmid back into that bacterium. Now that bacteria is going to divide through mitosis, asexual reproduction, but the plasmids also are going to divide. So you can see that as it divides, more and more plasmids with the isolated gene, we're getting more and more of that gene, are going to be found, and then we can pull it out and we can use it. So the production of insulin is done like this in order to mass produce insulin at high levels. I forgot to click my buttons, guys. There we go. So there's all the steps. All right. This video is about CRISPR, and you have CRISPR notes that you've already done or you are going to do. What I would also like you to do is using the benefits and unintended consequences chart in your notes. You can add your CRISPR notes in here, or you can use the ones I've given you and add those in too. But essentially, you need to watch this video and understand the process of CRISPR and what it does. Now, CRISPR is kind of the new version, the new technology to a restriction enzyme. So if you could please watch this video to make sure you understand CRISPR, as it's going to be a part of our lessons. So gene therapy is not in your notes and you don't need to worry about it, but I do want to talk a little bit about it. Okay. Gene therapy is kind of where we are in the world right now and trying to look at, can we modify mammalian chromosomes, so like human chromosomes, to treat genetic diseases? Gene therapy is still pretty largely in its clinical stages. There's a lot of questions and a lot of ethical issues that rise due to gene therapy. And so what happens is it's not actively used, but it is it is used in some ways, but it's not used in other ways. So it's used sometimes to try and help treat genetic diseases, but in the world of modifying, for example, any sort of genes in a baby before a baby is born, we are not there. So gene replacement is administered to a patient with defective genes using a viral vector. So you can kind of see the process here. Disorders like sickle cell anemia, diabetes, cystic fibrosis, all of these have been investigated. Okay. And the idea is what you want to do is you want to collect stem cells and you want to use stem cells that are not differentiated, so they don't have the gene changed. And what you want to do is you want to isolate that, put it into the DNA sequence, and then hope that this stem cell will take up and create new and better cells or ways to fix the disease. So again, this is just the process that's in your notes that you can see. So again, it's taken out, cells are altered, we use the vector, we insert it, and we go that way. I'm not going to go into a ton of detail because this video gives you a little bit of a better understanding of what gene therapy is. So I'm going to let you also watch this. Again, no notes need to be taken, but please watch as it is important. And we are going to watch a video called Designer Babies when we get back from break. All right, ladies and gentlemen, thank you and have a great day.